Interphase, the step before mitosis or cell division. Interphase consists of three of its own separate phases, G1, S, and G2. First is the gap 1 or G1 phase. During this phase, the cell grows in size. It then goes into the S phase, which stands for synthesis. This is where, as the name partially suggests, the DNA is synthesized. Helicase, which is an enzyme, first unwinds the DNA strand, making two single strands. Two new identical double DNA strands are formed based off of these two DNA strands. DNA polymerase, which is another enzyme, binds nucleotides to each of the original single DNA strands. So now, there's a new double helix of DNA that's just like the original. Finally comes the third phase of interphase, gap 2 or G2. In this phase, the cell checks for any errors that may have occurred so far, in which case it will not move on to the next step, which is mitosis, where the cell actually splits into two identical cells for growth and replacement. If there are no such errors, it checks to make sure the DNA strands are complete. If they're incomplete, it repairs them. Overall, in G2, the cells grow more and make proteins and organelles. The next step is mitosis.